Researchers at the Korea Advanced Institute of Science and Technology have achieved a significant milestone in vision science: the first successful long-term regeneration of retinal neurons in mammals. This breakthrough offers hope for restoring sight to millions affected by retinal degenerative diseases, such as retinitis pigmentosa, unlocking the regenerative potential of the retina. Unlike certain fish and amphibians capable of regenerating retinal tissue, mammals lack this ability. In species like zebrafish, Müller glial cells can transform into various neural types to repair retinal damage. However, in mammals, these cells do not naturally reprogram in response to injury. The Korean research team focused on a protein called PROX1, which accumulates in Müller glial cells after retinal injury and suppresses their regenerative capacity. By inhibiting PROX1, they reactivated these cells' potential to regenerate retinal neurons. In mouse models of retinitis pigmentosa, this intervention led to sustained retinal regeneration lasting 6 months, marking the first instance of long-term neural regeneration in mammalian retinas. Implications for human vision restoration. This discovery paves the way for developing therapies aimed at reversing vision loss in humans. While further research is necessary to translate these findings into clinical applications, the study provides a promising foundation for future treatments targeting retinal degenerative conditions.